All right, today we're going to be looking at DNA and RNA extraction.
Okay.
And this is something that is like really fundamental to so much research that's going on.
Right.
But it's not something that we always stop and think about,
Yeah.
how it's actually done.
Yeah, it's one of those things that, it's so essential. It's like the foundation of a house.
Right.
You don't see it,
Right.
but everything relies on that.
Exactly.
So without extracting DNA and RNA, you can't really analyze genes or study diseases or even develop therapies.
Right.
So it's really the first step to unlocking these secrets.
So it's really the bedrock of a lot of research.
Yeah.
And where do we even begin with this process?
Well, it all starts with breaking open the cells to release that precious cargo inside.
Okay.
The DNA and the RNA.
Right.
We call that cell lysis.
Okay.
And it's basically disrupting the cellular barriers to get to the good stuff.
So if you have a tiny cell,
Yeah.
it's like cracking it open like an egg to get to the yolk,
Exactly.
which is the DNA or RNA.
Yeah, and just like you wouldn't use a hammer to crack an egg.
Right.
We have to use the right tools for the job.
Okay.
So detergents can dissolve those cell membranes,
Okay.
kind of like dish soap breaking down grease.
I see.
Or we can use enzymes like proteinase K,
Uh-huh.
which act like tiny molecular scissors that snip away at the proteins,
Okay.
holding the cell together.
So you got your detergents and your enzymes.
Yeah,
But I'm guessing the process is a little different depending on if you're after DNA or RNA.
you're absolutely right. RNA is a lot more delicate.
Okay.
It's like a fine silk thread that can easily break.
Uh-huh.
It's vulnerable to these enzymes called RNases,
Okay.
and these are present everywhere they can be on our fingertips.
That's why they say on those crime shows, don't touch anything.
Exactly.
Because you can contaminate the evidence.
You don't want to contaminate anything.
Yeah.
So to protect the RNA from these molecular shredders, we use these special molecules
Okay.
called RNase inhibitors. And they act like tiny shields protecting that RNA during the extraction process.
I see. So it's like a very delicate dance you have to do
Yeah.
to get in there and get what you need.
Mhm.
So once you've broken open these cells, then what happens?
So now we've got this soup of all the components,
Right.
including our target DNA or RNA.
Uh-huh.
And so the next step is purification,
Okay.
which is basically getting rid of all the unwanted stuff that could interfere with anything downstream.
So it's kind of like separating the wheat from the chaff.
Exactly.
Yep. Yeah.
You're refining the raw material.
Okay. And so are there different methods for DNA versus RNA extraction?
There are, yeah. For DNA extraction, one of the classic methods is phenol chloroform extraction.
Okay.
And it's considered the gold standard because
Okay.
it yields highly purified DNA.
Right.
But it involves using these organic solvents that are kind of hazardous.
So it works really well.
Yeah,
But it's potentially dangerous.
right.
So are there safer ways to do it? Are there faster ways to do it?
Definitely. So silica membrane-based kits,
Okay.
they're a popular choice, and they kind of work like a Poffy filter,
Uh-huh.
allowing the DNA to bind while other components pass through.
I see.
And for large-scale projects, you might use magnetic bead-based methods.
Oh, wow.
And that's, imagine tiny magnets attracting and capturing that DNA
Okay.
so you can easily separate it from the rest.
Wow, that's amazing how they've developed all this.
It is?
So we've broken open the cells.
Yeah.
We separated the DNA. What's next?
Now it's time to address the elephant in the room.
Okay.
RNA extraction.
Right.
Remember how we said RNA is super delicate?
Yeah.
This makes the extraction process a lot trickier.
Because if anything contaminates it, it's ruined.
Exactly. Even the slightest contamination can just ruin the entire sample.
Wow.
So one method is to use this trizol reagent.
Okay.
And this separates the DNA, the RNA and proteins into these different layers
Okay.
based on their solubility.
Uh-huh.
So imagine shaking up like a bottle of oil and water. They eventually separate. So
Yeah.
you get these distinct layers.
Right. So you can isolate what you need.
Exactly. And then another method is to use these silica-based columns.
Okay.
And these are specifically designed for RNA.
Okay.
And they use these specialized buffers that stabilize the RNA and protect it from degradation.
So like giving it a coating or something?
Precisely.
Yeah.
But even with these methods, the key to successful RNA extraction is maintaining an RNase-free environment.
Right.
So think surgical levels of cleanliness here.
Oh, wow.
Every single reagent, every piece of equipment has to be free of RNases.
Yeah.
Or that RNA will be toast.
So no pressure.
No pressure at all.
So after all this careful extraction, then how do we even know if we got what we wanted?
Right.
How do we assess the quality?
Well, that's where quality control comes in.
Okay.
We can't just assume that everything went perfectly.
Right.
We need to check both the quality and the quantity of our DNA or RNA. It's like double checking a recipe before you pop that cake in the oven.
Right.
You want to make sure you've got the right ingredients and the right amounts.
So what are some of the tools that you use to do that?
Well, one of the first things we do is use a spectrophotometer.
Okay.
And that measures how much light passes through the sample.
Mm-hmm.
And this helps us to determine the concentration and the purity of the DNA or RNA.
So you can tell if it's contaminated with anything.
Exactly.
Okay.
We can see if there's any leftover proteins or anything else in there.
Okay.
And then another technique is agarose gel electrophoresis. And we basically run the DNA or the RNA through a gel matrix.
Okay.
Using an electric current.
Okay.
And this separates the molecules based on size.
Right.
And we can visually inspect the integrity of the DNA or RNA.
So you're looking for nice clean bands or something?
Exactly. We're looking for those nice clean bands.
Yeah.
That indicates intact DNA or RNA.
And what if you see something funky going on there?
Well, that's a red flag that something might have gone wrong during the extraction.
Okay.
So it could be contamination, degradation, or even a problem with the extraction method itself.
So you got to go back and figure out what happened.
Exactly. And for even more precise quantification,
Okay.
especially when we're dealing with really small amounts of nucleic acids, we use these fluorometric methods, and those use fluorescent dyes that bind specifically to the DNA or RNA.
So you have a lot of different ways to double check your work?
We do. We have multiple tools to make sure we're working with the top-notch material.
So you've got this top-notch material now.
Yes.
But what about things that could go wrong? I mean, this seems very complicated.
It can be, yeah.
So even with the most meticulous technique, you can still run into some issues.
Definitely.
What are some of the common ones?
Well, sometimes the yield is lower than expected,
Okay.
meaning we didn't extract as much DNA or RNA as we hoped.
So what might cause that?
Well, it could be due to inefficient cell lysis. Maybe those cells were a bit tougher than we anticipated.
Right.
And our method didn't fully break them open. Or we might have lost some of the DNA or RNA during the purification steps.
So like a leaky faucet, you're losing stuff along the way.
Exactly. And then there's the ever-present threat of degradation.
Right.
Especially for RNA. Remember those RNases lurking everywhere?
Yeah.
Even a tiny amount of contamination can lead to significant degradation and then make our RNA unusable.
And what about just contamination in general?
Contamination, yes, it can rear its ugly head in many forms. So leftover phenol or chloroform from the DNA extraction, or residual proteins, or even tiny bits of other nucleic acids.
Wow.
All of that can wreak havoc.
So it really comes down to being very careful.
It does. Attention to detail is crucial every step of the way.
Yeah. And sometimes even when you think you've done everything right, things can still go wrong.
Absolutely. And that's science for you.
Right.
Always keeping us on our toes.
So this has been really fascinating to go through this process of DNA and RNA extraction.
Yeah.
And I think it's really interesting to think about this as like the foundation of so much research.
It is.
What would you say are some of the key takeaways you want listeners to remember?
Well, first and foremost, I want to emphasize that DNA and RNA extraction is not a one-size-fits-all. The method you choose is really going to depend on what type of cell you're working with, the application you're using it for, and the level of purity that's required.
So you really have to understand what you're doing and why you're doing it.
Exactly.
Right.
And secondly, I can't stress enough the importance of quality control.
Okay.
Taking the time to assess the quality and quantity of your material can really save you a lot of headaches down the road.
It's better to be safe than sorry.
Exactly. And then finally, don't be afraid to experiment and troubleshoot.
Okay.
Even experienced researchers, they encounter challenges.
Uh-huh.
And the key is to learn from those setbacks and constantly refine your techniques.
So always be willing to learn new things.
Always be willing to learn.
Yeah.
Yeah.
So be curious.
Be curious.
All right. Well, this has been great.
It has been fun.
We'll have to continue this another time.
Definitely. It really is. Yeah.
So speaking of discovery.
Yeah.
Let's shift gears a little bit.
Okay.
And talk about some of the downstream applications.
Okay.
So once you have this high quality DNA or RNA.
Right.
What are some of the things you can do with it?
Oh, the possibilities are vast. It's like opening the door to a whole new world of molecular exploration.
Right.
So one of the most common applications is PCR.
Okay.
Or polymerase chain reaction. And this technique allows us to amplify specific DNA sequences.
Okay.
And make millions or even billions of copies from a tiny starting sample.
So it's like a molecular photocopier.
Exactly.
Making tons of DNA from a little snippet.
Exactly. And PCR has really revolutionized so many fields.
Like what?
Genetics, forensics, diagnostics. You know, it allows us to detect genetic mutations, identify pathogens, and even trace ancestry.
Wow, that's amazing.
It is.
It's amazing how much information you can get from just a small amount of material.
Yeah, and PCR is just the tip of the iceberg.
Oh, really?
Yeah, another powerful technique is DNA sequencing.
Right.
And this allows us to determine the exact order of nucleotides in a DNA molecule.
So you're reading the blueprint of life.
Exactly. We're reading the blueprint of life itself.
And what kind of insights can we gain from that?
Well, for starters, it allows us to compare DNA sequences between individuals, populations, or even species.
Oh, why?
So this can help us to understand evolutionary relationships, track disease outbreaks, and even identify genes that are associated with specific traits or diseases.
So it's really a powerful tool.
It is. It's like having a molecular magnifying glass that lets us zoom in on the tiniest details.
And what about RNA sequencing? How is that different?
So RNA sequencing is also very powerful. But instead of looking at the DNA blueprint, it focuses on the RNA transcripts, which are essentially the messages that the DNA sends to the cell's protein-making machinery.
I see.
So by analyzing these transcripts, we can get a snapshot of gene expression.
Okay.
So which genes are turned on or off in a particular cell or tissue.
So you're seeing how the DNA is interacting.
Exactly. We're eavesdropping on the conversation between the DNA and the cell.
And this is really important for understanding disease and stuff, right?
It is, yeah. This information is really valuable for understanding how cells function, how they respond to their environment, and how diseases develop.
Okay.
So RNA sequencing is being used to study everything from cancer to infectious diseases to developmental disorders.
So it has a lot of applications.
A lot of applications.
And beyond PCR and sequencing, are there other applications that use this extracted material?
There are, yeah. Another exciting area is microarray analysis.
Okay.
So imagine a tiny chip that's studded with thousands of DNA or RNA probes. And each one of these probes is designed to bind to a specific gene. So we can use this chip to analyze gene expression patterns across an entire genome. Or detect genetic variations, like SNPs, which are single nucleotide changes in the DNA sequence.
So it's like a molecular fingerprint.
Exactly. It's a molecular fingerprint that reveals these unique genetic signatures.
Wow. And so what are some of the real-world applications of that?
Microarrays are used in a wide range of fields, from drug discovery to disease diagnostics to personalized medicine.
Wow.
Yeah, and they can help us identify potential drug targets, predict an individual's response to certain medications, or even diagnose genetic disorders.
That's amazing.
It is.
And I know we talked about this a little bit before, but it's probably worth emphasizing that having really good quality DNA and RNA is essential for these downstream applications.
Absolutely, absolutely right.
What are some of the consequences if you don't have good quality?
Well, it's like using a blurry photograph to try to identify someone.
Right.
The results are going to be unreliable.
Yeah.
So if your DNA or RNA is contaminated or degraded, it can lead to inaccurate PCR results, faulty sequencing data, and misleading microarray analysis.
So all that work you did to get it is wasted.
Exactly. All that careful work we put into extraction is basically for nothing.
Wow.
If the material isn't top-notch.
And this is really important in some fields, like you're saying, diagnostics and forensics.
Especially in diagnostics and forensics, where accuracy is paramount.
So it really emphasizes that every step of the scientific process is important.
It does, every step from the initial sample collection to the final analysis.
So before we wrap up,
Yeah.
I'm curious to hear your thoughts about the future of this field.
Okay.
What are some of the advancements that you see on the horizon?
Well, the field is constantly evolving, and I'm particularly excited about the development of new, faster, and more efficient extraction methods.
So making it easier and faster to get what you need.
Exactly. Streamlining the process, making it more accessible to researchers.
Okay.
And with the rise of personalized medicine, there's a growing demand for these rapid and reliable methods that can be used in clinical settings. So imagine a future where doctors can quickly analyze a patient's genetic information to tailor treatments or predict disease risk.
So it's like science fiction becoming reality.
It is. It really is. And another area that's pushing the boundaries is single-cell analysis. So instead of extracting DNA or RNA from a bulk sample of cells, we can now isolate and analyze the genetic material from individual cells.
Oh, wow.
So this allows us to uncover this incredible detail about cellular heterogeneity. So the fact that not all cells in a tissue are the same, and how this contributes to disease development.
So like looking at one pixel in a giant image.
Exactly. And with advances in sequencing technology, we can now sequence the entire genome of a single cell.
Oh, wow.
So this opens up all these possibilities for understanding how diseases develop at the cellular level and for developing new, more targeted therapies.
That's amazing.
It is. And I'm also really fascinated by this emerging field of liquid biopsies, which involve analyzing the DNA or RNA that's circulating in bodily fluids.
Oh, wow.
Like blood or urine.
Interesting.
Yeah. So this could really revolutionize disease diagnostics. Allowing us to detect cancer or other diseases at a very early stage just by taking a blood sample.
Wow. So less invasive.
Exactly. It's like having a window into the molecular workings of the body without needing these invasive procedures.
And you could do that with both DNA and RNA.
We can, yeah. And the potential applications are vast. From cancer screening to monitoring treatment response to even predicting disease recurrence.
So this has really been remarkable.
It has.
To go through all this and think about how far we've come with DNA and RNA and how much information we can get.
It's an incredible time to be involved in science. And I can't wait to see what new discoveries emerge.
Yeah, it's really exciting to think about what the future holds.
It is, yeah. It really is amazing.
It makes you think about just how much information is packed into those tiny molecules.
I know, it's like they hold the key to unlocking the mysteries of life.
So let's zoom out a little bit and think about the bigger picture. How has this ability to extract DNA and RNA shaped our understanding of the world around us?
Well, for starters, it's completely revolutionized the field of medicine. I mean, think about genetic testing. We can analyze a person's DNA, identify mutations that can make them predisposed to certain diseases, and then allow for early intervention and preventative measures.
It's like a crystal ball.
In a way, yeah.
And you can kind of see what might be coming.
Yeah, and RNA extraction is equally important. You know, it allows us to study those gene expression patterns, which can give us insights into disease progression, drug response, and even the development of new therapies.
So it's not just about the genes themselves. But how they're actually functioning.
Exactly how they're being expressed in different cells and tissues.
And this is helping to develop new treatments for a lot of diseases.
Yeah, for a wide range of conditions.
And this has also been really helpful in the fight against infectious diseases.
Oh, absolutely.
If you can understand the genetic makeup of something, you can figure out how to stop it.
Exactly.
Yeah.
You know, by identifying the genetic makeup of a pathogen, we can track its spread, develop those diagnostic tests, and design those targeted vaccines.
It's like having a whole detective kit.
It is. It's a molecular detective toolkit.
And DNA and RNA extraction is being used in agriculture too, right?
Yes, it is.
You can improve crops.
Yeah, we can analyze the genetic makeup of crops and develop those varieties that are more resilient, that are more productive, improve the nutritional content, and even engineer resistance to pests and diseases.
It's amazing what you can do.
It is.
And then there's forensics, which has been completely revolutionized by DNA profiling.
Yeah, DNA profiling has helped to solve so many crimes, exonerate innocent people, and even identify missing persons.
It's amazing to think how much of an impact these tiny molecules have.
I know, it really highlights the power of science.
Yeah, and the ingenuity of the researchers who develop these techniques.
Absolutely. And the most exciting part is that we're really just scratching the surface.
Yeah.
Who knows what new discoveries are out there?
Right. It's a really exciting time to be alive.
It is.
And I hope that our listeners are leaving with a sense of wonder and curiosity.
Yes.
Because the world of DNA and RNA is just full of mysteries.
Absolutely.
So keep exploring, keep questioning, and never stop learning.
That's right. The future of science is in your hands.
Well, this has been really great.
It has been a lot of fun.
Thanks for joining us, and until next time, stay curious.